Hello, this is Renzo Huber, and I'm giving this presentation in context of the Minnesota Workshop 2019 at CMRR in November. And I want to talk about layer-dependent fMRI, specifically layer-dependent fMRI with sequences that are sensitive to cerebral blood volume changes. And as it has been shown a lot in the literature already, it's not so straightforward to reliably measure layer-dependent fMRI. The MRI method needs many quality measures to be fulfilled. While there have been a lot of advancements in the recent years to make layer-dependent fMRI more sensitive, more specific, have a larger coverage, faster acquisition, and very high resolution, it's not so straightforward to achieve all of these features at the same time. So in this presentation, I want to talk about our approaches to push as many of these pentagonal sequence parameters here at the same time. And I would like to talk about specificity and sensitivity first, because those are some of the most important challenges for layer-dependent fMRI. And as it has been shown already by many people in the field, simply increasing the resolution is not enough. In fact, when you go from 3 mm to 2 mm to 1 mm or open 7 mm resolutions, you still end up with these kind of fuzzy blobs. And it has been very well described already that this is coming from the fact that most of the oxygenation changes that conventional gradient echo bolt is sensitive to is coming from these vessels that are not as nicely laminar aligned like the neurons are. So in some sense, using conventional gradient echo bolt as a measure of layer-dependent activity is like using a CO2 emission mapping tool as a measure of industrial activity. It might work very well to compare the big structures, the countries or even the cities, but it certainly breaks down as soon as we approach the spatial regime of the training infrastructure. And we can obviously not use the overall CO2 emission content of a voxel like that as an indication of the activity in the scanner building over here, because most of the CO2 emission might come from cars going to other buildings somewhere else. Similarly, here most of the oxygenation changes in the superficial layers are probably coming from neural activity further upstream. And this has been well described, and there are alternative methods already proposed that are being supposed to be more locally specific. For example, this blood volume sensitive VASO method, which can better delineate these stripy patterns indicating the layers. However, it also comes along with a lot of weird stuff. So we have these false positives here. There's a higher noise level that comes along if you want to have this higher localization specificity. And one very efficient way in increasing the SNR is to move to higher and higher field strength. While clinical field strength of 3 Tesla just barely exceed the detection threshold at these resolutions, 7 Tesla is already a whole different world, and you can better delineate these stripy patterns. You might even get better sensitivity with even higher field strength. However, it also comes along with a lot of technical challenges. And I believe you can think of it in this metaphor, where 3 Tesla is the most cost-efficient way of doing things, whereas 7 Tesla is more expensive, but it gives you these extra performance. And you might be able to break records at these higher field strengths if you're willing to work with these prototype tools and you have a whole set of, like a whole team of engineers working on it, helping you, which is not always the case if you're a postdoc. So with this, we can come back to this pentagonal sequence parameter space and conclude that VASO allows us to improve the localization specificity compared to conventional gradient echo bolt methods. However, this comes at a price of a lower sensitivity. And obviously, sensitivity and specificity are not completely independent measures, and the best specificity in the world is useless if you don't have the sensitivity to measure anything, or vice versa. And the best sensitivity in the world is useless if you don't know where the signal is coming from. And as many people previously in the literature have shown, specifically Kamil Uluda, the most of the sensitivity in gradient echo bolt is actually sensitivity that we don't want to have. 90% or so are coming from the extravascular bolt of large vessels, and only 10% are actually locally specific signal changes. And you can get the right neuroscientific answer out of gradient echo bolt in layer-dependent fMRI if you kind of throw away these 90% and use very clever task designs, contrast different task conditions, or use the vascular deconvolution model from Camille, which basically effectively removes the 90% and then the 10% can save the day. So it might make sense also to use a method with a lower sensitivity, but then where you don't need to throw away 90% of the signal. But we should also not ignore the acquisition parameters down there of coverage, TR, and resolution, where we always want to push them all at the same time. But as soon as you push one of them, the other ones need to be sacrificed somehow. Most of the cases. But every now and then in the field, magic happens, and the field comes with a new advancement that allows us to push more of those. And while this is mostly the case for hardware improvements, I want to focus mostly on software magic here and sequence improvements to push these acquisition parameters all at the same time with less compromises. So let's start with the coverage. How can we increase the coverage in VASO? VASO is an inversion recovery method where we have a T1 weighting to make it sensitive to blood volume. And we usually acquire the signal right at the blood nulling time here, which means that we have a very small acquisition window and can acquire a few acquisition slices only. We can kind of extend this a bit by ending up with similar T1 values to get a few more slices, but the ultimate physical limit is in the range of 2 seconds. Beyond that, T1 is just too short and the signal decays too fast. So we are working on alternative methods, such as this MAGIC method, which stands for multiple acquisition with global excitation cycling, where we try to manipulate this recovery curve with variable flip angles that we manipulate across different 3D EPI K-space segments. So when you have a smaller flip angle, this inversion recovery curve is not free anymore, but we can kind of freeze the T1 weighting somewhere up there. With larger flip angles, we freeze the T1 weighting somewhere down there and can basically extend the T1 weighting as long as we want to acquire as many slices as we want. Like in this example here, where we acquire more than 100 slices with 0.8 millimeter isotropic resolution to cover the entire brain. And in the background, you see the mean VASO image, which has this inherent T1 weighting. And in the foreground, you see the common brain-wide network. So for example, in the second row here, you see the default mode network with its conventional like RIs of these 4 different blobs here. On the other hand, here we have the frontoparietal network, we have the visual network over here, and all these networks really benefit from having this large coverage and getting all the RIs in there at the same time. And you can better appreciate this maybe even on this figure here of the stills, where you can really follow the single layer activity in the default mode network in this brain area, whereas we have these kind of double stripes in the frontoparietal network, especially in the interparietal sulcus here or also here. So we do have layer-dependent signatures, and we just need to better understand how to interpret them and understand what they mean. But it definitely opens up a whole new level to investigate brain systems and connectomes. And usually in resting state connectomics, people divide the brain in different RIs and then look at area-to-area connections to end up with these connectivity matrices. Now with Layer-FMRI, we have an additional dimension here and can basically look at every single edge here and how this connectivity comes about from which layer. For example, in those nodes here in the default mode network, we see that the PCC is kind of unique, that most of the fluctuations are really dominated from the middle cortical layers compared to the other areas, which are much more driven from feedback activity with the superficial layer peak. And this tells us that these other areas are much more feedback driven. They're kind of being passively dragged along from the actual hub of the default mode network, the PCC. We can try to validate these kind of connectivity approaches and analysis with networks where we already know the functional hierarchy. For example, between V1 and V5, where we know that feed-forward input from V1 to V5 terminates in the middle and deeper layers, whereas the feedback input from V5 to V1 mostly terminates in the superficial layers. Or similarly also in the primary motor sensory areas, where we know that the primary motor cortex receives feedback from the sensory areas in the superficial layers only. And as soon as we have this validated, we can also go to new unexplored circuits, for example, in the frontoparietal network, where we see very clear layer features, such as in the interparietal sulcus, where we have these different fluctuations in the superficial layers and deeper layers as different clusters, which can be seen down here, often even as independent components, where one is in the superficial layers, the other one is in the deeper layers. So with this, we can go to larger coverage, even whole brain. But what about TR? So if we want to shorten the TR, again, we can benefit from this magic approach because it doesn't really necessarily need this inversion pulse, but just a variable flip angles to get the T1 weighting. And when we sacrifice coverage, we can definitely go to sub-second TRs. And this is a real-time movie here that you see where we have a very fast sampling rate of new image, new image, new image, new image. This is the rate that images come in. And this allows us to really follow the hemodynamic response across these two stripes of different layers in the motor cortex for bold and vaso, especially in this post-stimulus period over here where they differ quite a bit, if you care about those features. In reality, it doesn't really matter so much to have whole brain coverage, and you already also don't really need these sub-second TRs. Usually the task is much slower anyway. And in reality, when neuroscientific collaborators approach us, they come along with very specific hypotheses about a certain brain area. And in those cases, it can be beneficial to use a slab acquisition with a few second TR and then use all the sensitivity that we have and invested in very high resolutions. And there we need to use different approaches, especially when we want to get to like 500 micrometer resolutions, because there a single shot EPI, even in 3D EPI, doesn't really cover the entire matrix with one shot. T2 star is just too short. So we need different approaches, such as a segmented readout, where we have faster shots and then multiple of those that are concatenated. And this is something where we collaborate with the Bonn group, specifically Rüdi Stirnberg, who have a 3D EPI sequence that allows us to segment within and across partitions. And we use that sequence to segment across different vaso TRs after the inversion pulses, which then allows us to choose matrix sizes as large as we want with the same kind of same sampling rate that comes in. And as always, we see that in vaso, we have a very specific activity following the tree of Gennari in this case, whereas bold is much more sensitive. We have many more significant voxels compared to vaso. However, most of them are actually coming from areas that we don't want to see, like above the cortex. And this is also highly consistent across participant, where we always see these nice outlines following the stria here. So with all these various sequence approaches, we have a lot of flexibility to move within this pentagonal parameter space. So in applications, depending on the neuroscientific research questions, we can push the sequence parameter space as needed. And in the remainder of my time, I would like to show you a subset of experiments that we did. For example, in the motor cortex, where we really pushed for sensitivity, sensory cortex, where you need more coverage and then resolution, more cognitive approaches in the motor cortex, visual areas, working memory areas, and associate cortices all across the brain. So let's start with the motor cortex, where we have very clear hypotheses about the densely packed microcircuit from animal tracer studies, namely that the superficial layers send a lot of feedback into receive a lot of feedback from other cortical areas, like sensory feedback, whereas the deeper layers are the ones that triggers the muscles to move. They have the corticospinal connections, and we can try to manipulate these different circuits with different tapping or touching tasks. So for example, for a tapping here, the muscles are moving, so we do expect activity in the deeper loops, but there's also sensory feedback because the fingers are touching in the superficial layers. So we actually do expect superficial and deeper activity visible as two peaks here in blue. Whereas for the touching condition, the participants are asked not to move their fingers, and therefore we only see this feedback from the sensory cortex, from the passive touch, but there's no output, no muscle movement. And recently we also pushed this more to the cognitive domain using imaginary tapping in collaboration with Andrew from Alex Martin's lab. And there we not only ask the participants to do this tapping movement, which again gives us this double peak, but also imaginary tapping in red over here, which does not have the output because there are no muscle moving if you just think about moving your finger. But still in the same position of the tapping, we have the superficial peak over there, which you might be able to see right at this part of the motor cortex. And it actually matters where you look along the motor cortex. And I learned that you cannot actually do layer-dependent fMRI without knowing where you are in the columnar domain. So we need some topographical tools always when we want to do layer-dependent fMRI. And I developed those again in the toy model of the motor cortex, which I want to show you here in this kind of axial-like view of the central sulcus. And on the left side, we have kind of a topographical space now emerging for the sensory cortex in black. And on the right side, it would be the motor cortex in white. And all this is done completely in the voxel space of EPI, so there's no resampling necessary between like vertices and voxels to get towards this topographical representation. And this allows us also to work in 3D here, and rotating it to look at it from the backside of the brain, where this would be the midline of the brain, this would be the right ear over here, and we can project the infrared fingers onto the sensory cortex here with pinky finger, ring finger, middle finger, index finger, and thumb. And in the sensory cortex, as expected, we have this somatotopical alignment of neighboring fingers being represented neighboring in the brain. And it's also pretty huge. Like a lot of the central sulcus is really busy representing these individual fingers. More interesting is then what happens on the motor side. So when we switch this around and look at the hand knob, which would be this cave over here, and project the individual fingers of the pinky finger, the ring finger, we actually have 2 ring fingers, we have 2 middle fingers, we have 2 index fingers, and 2 thumbs. So in the motor cortex, it's really more complicated. It's much, much smaller than the sensory cortex. So here, this entire somatotopical hand representation is barely exceeding 3 to 4 millimeters. So you only see this with very high resolutions. But we also have these mirrored patterns that is better visible maybe if we look at it in a projection along this plane, where you see on the bottom left that we have one linear representation of the sensory cortex, and on the top right in the motor cortex, we have multiple mirrored representations. And those patterns are also highly consistent across participants. And the different patches in the motor cortex suggest us that the motor cortex is not really a body part map, but more an action map. And these different patches are differently engaged for grasping versus retraction movement. So it matters if you move your one finger in one direction or the other direction. Different muscles are involved. We can also use layer-dependent fMRI in more cognitive tasks, like in this example here in the dorsolateral prefrontal cortex, where we also have very specific hypotheses about the densely packed microcircuit. For example, we know that we have cue cells, we have delay cells, and response cells that eventually trigger the motor system, differently aligned across the different cortical laminae. And Emily Finn here, my collaborator, she found an old task to engage these different cells differently. For example, by showing participants these strings of letters and then cueing them to either memorize the letters in the order they saw them or sort these letters according to an alphabetical order in their mind, which is then believed to engage mostly the superficial layers. Later in the task, the participants are asked to do a response decision, which is then believed to mostly engage the deeper output layers. And this simplified microcircuit really nicely explains all the variance that we are seeing in our data. Here, for example, for the response condition, we see activity mostly in the deeper layers following the cortical ribbon, whereas parallel to that in the superficial layers, we have this alphabetization activity, which might be better visible also looking at the time courses, where you see that the superficial layers, they really shoot up as soon as the participants start with this mental alphabetization task. And as soon as they found a solution, it goes back to baseline. Whereas for the deeper layers, they don't really care much about the alphabetization, and they only shoot up as soon as you need to do this response decision. We also tried to do layer-dependent fMRI with blood volume imaging in the sensory cortex in the context of predictive coding. And in the sensory cortex, we know that most of the feed-forward thalamic input comes into the middle layer 4, whereas expectation or predictive error calculations come back into the superficial feedback layers. And we can try to engage these different circuits with different touching tasks. So in one condition, the participants learn specific sequences of being touched, and then in some instances, we leave out individual fingers here. And this blue condition really is expected to enter in the superficial layers. Whereas for one of the many control conditions in green here, participants are asked to predict which finger is going to be touched next, but it's a completely random order. So they cannot predict which finger is going to be touched next, but there's actually feed forward happening on the index finger, which is then visible as 2 different peaks in the layer-dependent blood volume profiles. For example, the blood volume has this clear superficial peak for the prediction activity in blue, whereas a clear middle feed-forward peak for the feed-forward thalamic input. And with this, I want to thank all my colleagues, and I thank you for your attention.